Were you guys trying to get into the meeting? Because when I got in the meeting, there was nobody there, and I logged out and re-logged in, and now there's six people here. So I'm just wondering, what's the story?
I've been in the loading screen since 2:30 and you just popped up.
All right, I don't know what happened because I was in Zoom, supposedly the first lab, and nobody was there, and so that's why I logged out and re-logged in. Anyways, I'm glad people are here. I'm not sure what's going on. I did have a little bit of trouble with Canvas. Um, but that shouldn't affect Zoom, so I'm not sure what's going on. Anyways, before we begin, are there any questions? Let me share my screen here. I can get out of this. We got two of them open here because I had one open earlier. Let's go to the lab. Well, let's go to the syllabus. So today is November 4th, and we are supposed to be doing Lab 8, identification of gram-positive organisms. Any question about any of that? Let me state that there was something that rolled over incorrectly from last term, and it had the quiz in the wrong date. I've now corrected that. It's following the date on the syllabus. We're having the quiz not this week, but next week, and that'll be quiz 4. I do have the date correct in the Canvas. I think it was in the, not in the, not on my main web page. It was correct on my main web page, but in the, oh, what is it? The grade book. It had the quiz on the wrong date. So if you see an error like that, let me know and hopefully I'll get it corrected as soon as possible. Um, all right. Any questions about what we're doing? Blow that up a little. Let's move on to lab 8. Lab 8. Let's blow this up a little too. Lab 8, identification of gram-positive organisms. We're going to be talking about gram-positive cocci, Staphylococcus, Streptococcus, Enterococcus, and then gram-positive bacilli. Sorry, this table's gotten messed up. Clostridium, family Clostridium, family Bacillus, Corynebacterium, and Listeria. Those are the gram-positive bacilli. Any questions about any of that? All right, there are some video clips, supplemental materials in this lab. There's some gram-positive dichotomous key and some case studies. These last two are provided as separate files in Canvas. Let me see if I can show you that. I don't have it open anymore. We must be in week 7 lab. Boy, we're already in week 7 of the class. So there's the lab module, and there's the worksheet for lab 8. And then there's 8 case studies, one after the other, and a dichotomous key. Only use this dichotomous key for this lab. Don't use it for your unknown project or any other labs. Only for this lab. The dichotomous key is a dichotomous key for identifying species of bacteria. And on the first page, we're identifying certain of the gram positives. See if I can scroll down here. We're going to be following the gram-positive cocci. We'll talk about the gram-positive bacilli in a minute. So you can split the gram-positive cocci into those which are catalase positive and those which are catalase negative. We're going to say scratch out the catalase positive, our unknown is catalase negative, and then you do the hemolysis test. This is alpha hemolysis, beta hemolysis, gamma hemolysis. Sorry, the hemolysis isn't up here, but that's what that means. Of the alpha hemolytic bacteria, there's only two that we're going to look at, Streptococcus pneumonia and Viridian Streptococcus. And you can separate these two by doing the optogen test. If it's optogen positive, it's Streptococcus pneumonia. And if it's optogen negative, it's Viridian Streptococcus. For the gamma hemolytic bacteria, we're going to be talking about two, Enterococcus and Streptococcus. And for these, you need to run two tests, the bile-esculin test and the SF broth test. If the bile-esculin and SS broth are both positive, then it's Enterococcus faecalis. And if it is bile-esculin negative and SF broth negative, it is some gamma streptococcus. We don't identify it further in this dichotomous key. And then if it's beta hemolytic cocci, then it's either Streptococcus pyogenes or Streptococcus galactiae, and you can separate them by doing the base tracing test and the CAMP test. If it's base tracing sensitive, meaning positive, and CAMP negative, it's Streptococcus pyogenes. If it's base tracing negative and CAMP positive, then it's Streptococcus galactiae. Any question about using this key or this lab? Now, we do have gram-positive bacilli, and you see it goes there. You go to the next page to follow the gram-positive bacilli. So the gram-positive bacilli, you can separate them into anaerobes and aerobes. If it's an anaerobe, we're only looking at the Clostridium species, Clostridium family. If it's an aerobe, we then separate them. Sorry, I lost my page here. We then separate them on, I'm getting a hard time getting that. We then separate them into whether they have an endospore or not. If they make an endospore, it's a Bacillus species. And if it doesn't make an endospore, it's of one of two families. And I'm not sure why I can't show you that. Let me shrink this down and then maybe I can show it to you. There we go. It's endospore negative. It's either Listeria or Corynebacterium species. You can do the hemolysis test, and if it's gamma hemolysis, it's Corynebacterium. And if it is not gamma hemolytic activity, but beta hemolytic, and then it has a CAMP positive, although that's a weak positive, it's Listeria monocytogenes. So we actually identify the species. Any question on the dichotomous key here? All right. And let's come back to here. I thought about putting all of these case studies and then one file, and then you'd only need to access one file. But I'm glad I kept it separate because this is the way well, he's now the department chair, but the lead microbiologist sent them to me. And I'm glad I kept them separate because he changed, I think it was these two, case study 4 and case study 5. And so I'm glad I kept them separate. Then I needed only to change these two files, which he sent to me. So I just needed to upload them instead of redo the whole thing. Any question about any of this? Remember, your infectious disease research project is due November 8th. That is this Saturday. And why didn't I see that on the syllabus here? I just didn't read it. Unknown work, infectious disease paper is due this Saturday. If you haven't picked a topic, make sure you okay your topic with me first, or you'll get a 10-point penalty. You were supposed to be doing an infectious disease. I'll check on that. I break some of the diseases up into, I don't know, I break some of the diseases up like the flu. I break it up into influenza A, influenza B, influenza C, so I can break it up into different species or groups. Some of them I break the disease up into later stage disease like Streptococcus pyogenes or early stage disease. And that's to allow multiple students to choose that disease. Any question about anything? All right, let's go on with the lab module. The learning objectives. This isn't the right file. I don't have something highlighted here. Here it is. I got the table set up. Good here too. The learning objectives. 1, understand the principles and how to interpret the results for the CAMP test, the Bialis-Gulen auger slant test, the SF broth test, and the optogen sensitivity test. And then 2, be able to use a dichotomous key to solve case studies involving gram-positive bacterial infections. And let me warn you, you can use AI to solve these problems, but if you do, then when you're given a dichotomous key case study problem on the quiz or the final, you won't be able to do it because you haven't actually done it yourself, or you'll have a much harder time doing it, I should say. And so you really should do it yourself. And then if you like, you can check your answer using um, AI. But you should not use AI because you need the practice for solving the case studies, which you will find on the quizzes and the final. Any question about any of that? All right. When specimens are received in a microbiology lab for routine bacterial cultures, they're inoculated on culture media that will facilitate the growth of common pathogens and then placed in a physical environment that will support their growth. Microbiologists do use colony morphologies and the gram stain to provide important information about the bacterial isolates. However, further biochemical testing is required in order to identify these species. In this lab, we're going to talk about the CAMP test, the Bialis-Gulen hydrolysis test, the SF broth test, and the optogen sensitivity test. Any questions about any of that? All right. The CAMP test is CAMP is an acronym of the 3 microbiologists who discovered it, Christie, Atkins, and Munch Peterson. And they first described the amplified hemolytic effect that some beta hemolytic organisms have when they're grown near Staphylococcus aureus. What you do is you streak a single streak of Staphylococcus aureus, which is beta hemolytic, down the middle of a blood agar plate. And perpendicular to that Staphylococcus aureus streak, you make a streak of another unknown organism, which is beta hemolytic. So there is, let me blow this up so you can see even better. So right here is the streak of the cell Staphylococcus aureus, and you can see it's clear around it, and that's because of the beta hemolytic activity of Staphylococcus aureus. Your test organism, you then streak perpendicular to Staphylococcus aureus, and you look for enhanced hemolysis in the growth around the Staphylococcus aureus. And let me blow that up even further because Streptococcus pyogenes is really tiny in its growth. You can see the streak of cells, which is kind of orangish going this way. And then Streptococcus pyogenes is beta hemolytic. So there's a clear region around the Streptococcus pyogenes. There is no enhanced hemolytic activity around the growth of Streptococcus pyogenes near the Staphylococcus aureus. So Streptococcus pyogenes is CAMP negative. Well, I don't see that here, but just trust me on that. That's CAMP negative. Oh, there it is, negative CAMP. Listeria is a weak positive CAMP. So here we have our Staphylococcus aureus. There you have the Listeria. That region right there is the growth. And then there's a very thin beta hemolytic clearing around Listeria, which gets enhanced in this region of Listeria's growth near Staphylococcus aureus. So slightly positive CAMP test for Listeria. And then we have Streptococcus galactea. The streak is here. You can see the beta hemolysis of Streptococcus galactea here. And there you see a greatly enhanced hemolytic activity of Streptococcus galactea grown near Staphylococcus aureus. And normally you do get an arrow-like appearance when you have this CAMP test and it's positive. I'm not sure why it forms the arrow, but that's what it is. On this side, I know why it's not enhanced up here, and that is because the the factors giving the enhanced hemolytic activity from the Staphylococcus aureus are being released by the red blood cells, which are lysed by Staphylococcus aureus, and they're diffusing into the blood agar to only here. They don't diffuse further, and that's why we have the arrow like that on this side. But why it's like this, I'm not sure about. Any question about any of that? But you normally see an arrow-like appearance. If you look even closely, that's kind of an arrow. Anyways, that's the CAMP test, looking for enhanced beta hemolytic activity of a beta hemolytic organism growing near the Staphylococcus aureus streak. So CAMP test is only used on beta hemolytic unknowns. All right, let's talk about the bile esculin hydrolysis test. It uses a agar slant, and it's used to differentiate Enterococcus from Streptococcus, these two families of bacteria. If the organism can hydrolyze esculin, which is in the tube, it will make the product esculatin. A dark brown black precipitate will appear in the tube. So esculin is hydrolyzed to beta glucose. You don't need to know that, but esculatin is a very dark brown color. It really looks black. And to be positive, you should have 50% or more of the tube black like that. A negative is less than 50%, or most of the time it's no color change. The tube's the same color as what began. Streptococcus species are negative in the bile esculin hydrolysis test. Let me shrink that down. And Enterococcus species are positive in the bile esculin test. Any question about the bile esculin test? All right. The SF broth test stands for Streptococcus faecalis, which is an old name for what we now call Enterococcus faecalis. It was initially grouped in the family Streptococcus, but when they sequenced the DNA, they discovered that Enterococcus faecalis belongs in its own family of Enterococcus, and there's other species in the Enterococcus family. It is not in the Streptococcus family, but this test was invented a long time ago, and so we still call it the Streptococcus faecalis broth test, and I'll just call it the SF broth test so that nobody gets confused, because the bacteria is no longer called Streptococcus faecalis. It is called Enterococcus faecalis. Any question about any of that? Anyways, um, the SF broth test has a purple color. It has sodium azide and carbohydrate glucose in it, a pH indicator, which gives it this purple color. If the bacteria can hydrolyze the glucose, it'll make acid products, changing the appearance of the color in the tube to a yellowish color. So if the organism can ferment glucose, it'll be SF broth positive. If it cannot ferment glucose, it will be SF broth negative. Any question about any of that? Streptococcus species, all members of the family are SF broth negative. Enterococcus faecalis is SF broth positive. Any question about that? All right, the next test we're going to talk about is the Optogen sensitivity test. Optogen is a antibiotic, and it's used to differentiate between Streptococcus pneumonia and other alpha hemolytic streptococci. So what you do is you identify that it's a member of the family Streptococcus, and you streak it on blood agar. Let me blow this up so you can see it a little better. And if the bacteria have alpha hemolysis on the blood agar, let me blow that up even further. You can see the bacteria are whitish or off-white. I'm not sure why it's like a double colony, but that's the way it is looking. And then you see a darkening of the blood around the whitish or off-white colonies. And that is from alpha hemolysis. So we have a member of the family Streptococcus having alpha hemolysis. You then take the bacteria and streak it in this region of a blood plate, and then stick on a paper disc containing the antibiotic Optogen. If the bacteria are resistant to the Optogen, they will grow right up to the paper disc, and that is not Streptococcus pneumoniae, because Streptococcus pneumoniae is sensitive to the antibiotic Optogen. And you should see a zone of inhibition greater than or equal to 14 millimeters, and that is Optogen sensitive. If the zone of inhibition is less than 14 millimeters, we then classify it as Optogen resistant, down to 0 zone of inhibition, which is shown here. Any question about any of that? All right, let me briefly talk about actually, I've already talked about the dichotomous key. So you use the dichotomous key to follow dichotomous logic, where you have a bunch of species, and then you run a test which separates the species into one of two categories. You throw out the one that it's not, and then run more tests to separate the species that the first test was positive for. An example of a dichotomous key, we have all the species of bacteria. We do the gram stain, and if it's gram positive, we'll then take this one. If it's gram negative, we're going to throw these out. So it's gram positive. You can then split the gram positive bacteria into the gram positive bacillus and the gram positive cocci. We're going to throw out the gram positive bacillus and talk about the gram positive cocci. So we can take the gram positive cocci and run the catalase test on it. If it's catalase positive, then it is of family Staphylococcus. If it's catalase negative, it's not Staphylococcus and it's either a family Streptococcus or Enterococcus. We're going to throw these out and then follow the catalase positive, gram-positive cocci, and we can do the coagulase test. If it's coagulase positive, its species is Staphylococcus aureus. If it's coagulase negative, then it's some other member of the Staphylococcus family other than Staphylococcus aureus. And then you can do the novobiastin test to separate out among those species. Any question about the dichotomous key example? All right, let me go to the worksheet. You're not going to be physically performing any of these activities, but you will be held responsible for knowing how to perform and interpret them. We have the video clips discussing the SF broth test and the bile esculin test. We have another video discussing the CAMP test. So in the worksheet, the first thing to do, question 2, is to fill in this table. I put in the first table result for you. So the gram stain for cocci and bacilli, the positive result is a purple color, and the negative result for the gram stain is a pink color. For the Schaefer-Fulton stain for endospores, give me the positive result here and the negative result there. If you need more room, just expand the column and type it in here. You can also expand the table by blowing it up that way. For the negative stain for capsules, give me the positive result there and the negative result there. For the catalase test, when you add hydrogen peroxide to the bacteria, give me the positive result and give me the negative result. For the coagulase test, give me the positive result of when you're adding bacteria to rabbit plasma, and give me the negative result for coagulase. For the oxidase test, give me the positive result and the negative result. And this one I know is not discussed in this lab. You have to look at an earlier lab to answer some of these. I think that one was also done in an earlier lab. The gram stain is not done in this lab, but you should know it. If you don't know it, you can look it up in an earlier lab. The negative stain for capsules is also done in an earlier lab or in the textbook, a lecture. The CAMP test is discussed in this lab. Give me the positive result and give me the negative result. For negative, you can say something like no enhanced hemolytic activity, which is a bit large, but you can always say no enhanced hemolysis. And then for the bile esculin hydrolysis test, give me the positive result and the negative result. For the SF broth test, give me the positive result and the negative result. For the optogen sensitivity test, give me the positive result. And remember, there has to be a size for the positive result, meaning you have the diameter of the zone of inhibition has to be greater than a certain size. And then give me the negative result. For the basotrace and sensitivity test, give me the positive result and the negative result. And the basis trace and sensitivity test was discussed in an earlier lab. Any questions about any of this? If you need help developing and using a dichotomous key, you can see this link or click this link and then go through the example that's here. If you're perfectly comfortable with the dichotomous key, you can skip 3A. And remember, we have a dichotomous key file in the Canvas website. You need to open that, and we're going to be talking about these species of bacteria. and then open up the case studies for case study number 1. Give me the identification of the gram-positive bacteria. List the species name here. Usually it's a species name, scientific species name, but occasionally it might just be a family of bacteria. And just follow whatever the key gives. For case study 1, let's go ahead and open that one. Actually, rather than opening it in Canvas, let me go here. Case study 1, a 13-year-old teenager shown here attended a week-long summer scout camp. Upon returning home, his mother notices a pustule beginning to develop in a spider bite on his arm. The area is warm to the touch and becoming extremely painful. It's also hard. When her son starts running a fever, she takes him to a walk-in medical clinic for a physical evaluation and testing. And here are the findings. That is the gram stain. You have to blow that up to see that. So there's the bacteria here. These large pink cells are actually the boy's cells, but those are the bacteria causing the infection. And that's the infection inside the boy. And let me shrink it down now. There's the bacteria grown on a blood agar plate, and you can see um, something happening to the blood in that plate. And then they're doing the catalase test. Come on. Yeah. So they have the bacteria there that they got from the blood agar plate, and then they're adding hydrogen peroxide to it. And there's the result of the catalase test. And then for test 3, they're growing the bacteria on a McConkey's agar plate. The bacteria is shown on the left side of the plate. Is that McConkey's or mannitol salt agar? Let me take a look at this. Hmm. This is mannitol salt agar plate. Okay. Sorry, I said McConkey's because those two plates are similar, but this is a mannitol salt agar plate. And left side, it tells you it's negative for growing on the mannitol salt agar plate. And then we have the novobiocin sensitivity test, where we took the bacteria here and streaked it out on the plate and then put novobiocin there. And I'm not sure why that looks different. You just ignore the fact that that's looking different than the blood agar plate here. Maybe that's not blood agar, I don't know. And this obviously is resistant to novobiocin. And then give me what species was isolated and write it in case study number 1. Remember the species, if you use AI, it must be a species that is talked about in this lab, otherwise it is wrong. So Once again, you shouldn't be using AI, but you can if you wish. And just remember that if you use AI, you're not going to be able to use AI to answer a case study on the quizzes and on the final. So it would be better not to use AI. And then what you can do is once you get your answer, you can use AI to confirm your answer. Let's go to case study 2 because I need to talk about that right there. Come on. A 27-year-old female is receiving prenatal care. The physician orders routine vaginal culture. She is not exhibiting any signs or symptoms, so it's just a routine culture. And the Thayer Martin has no growth. Lactobacillus with other vaginal microbes were cultivated on BAP. However, there are several colonies of interest. These colonies were streaked for isolation on another blood agar plate. And here are the results. Get rid of that right there. You can see hemolytic activity there. And then there's the gram positive the gram stain of the organism. You need to blow that up to see the cells. So there it is there. We then have some rapid tests. This is the catalase test. Taking the bacteria here, putting on the slide, adding hydrogen peroxide. There's the result of the catalase test. We then do test 2, which isn't named in the lab module. So I've named it in the case study 2 on the worksheet. Case 2 is the basic trace and sensitivity test, which was talked about in this lab or an earlier lab. Where is the thing? Here we go. And there's the results of the basic trace and sensitivity test. And then we're doing the bacteria and the CAM test streaked here next to Staphylococcus aureus there. So get your results of the CAM test. And then what bacteria was isolated? Once again, you must give the species or the family that is named in the dichotomous key. Any question about any of these? All right, I'm not going to discuss the other case studies. You can go ahead and read through them. I will be here until 4 o'clock, or the last student is logged on. So if you have a question, make sure you ask early because once all students leave, I'm going to leave as well. Any questions?
I just have a question about 3B. It just says to review it. Are you expecting us to answer any sort of question with 3B?
Let me get to 3B. This is in the worksheet, correct? Yeah, there's
Yes.
no question on 3B. It's just these are the species of bacteria that could be one of the case studies because these are the bacteria we're talking about in this lab. There is no question to 3B.
Okay, thank you.
All right.
I had a question about my unknowns.
Okay.
So I sent you a message in Canvas, but I didn't know if you got it. Basically, I requested I think it was the nitrate reduction test. Um, and you sent me the results before adding zinc. I was just curious. I do need the results for after zinc too, don't I?
Um, Not really,
Okay.
because you should be able to determine the results for the nitrate reduction,
Mm.
and we're only answering nitrate. we're not nitrate. We're not trying to answer nitrite.
Oh, okay.
Okay. So
Okay, thank you.
it is true the test can do that, but I'm not giving you the results for the nitrite. So that's why I've only got it um, as stated, meaning it's only for the nitrate.
Okay,
Okay.
thank you.
All right.